All right, I'd like to introduce our first speaker. Today we are going to be joined by Dr. Claire Wade. She is a professor of animal genetics at the University of Sydney and chair of computational biology and animal genetics. She began her career in quantitative genetics before making the move to genomics in 2002, when she began a position with the Whitehead Institute for Biomedical Research at MIT. While in the USA, Claire worked on several mammalian genome projects, including the mouse, dog, and horse, for which she was the lead researcher. Claire's research interests include unraveling the secrets of the genome, biology using next-generation sequencing. In particular, she studies the application of new genomic technologies to improve our understanding of diseases and behavioral traits in domestic animals and wildlife, and our understanding of the links between DNA and phenotype in general. Please join me in welcoming Dr. Claire Wade.
Hello everybody and welcome to SPARCS 18 on the second day of the meeting. It's really exciting to be here and I'm very thankful to the organisers for inviting me to come and speak to you. I'm going to talk to you today about some active research that's happening in my laboratory. I have a broad range of interests in the lab. We work on everything from Tasmanian devils to Sumatran tigers to dogs and horses. But I'm going to talk to you today about some of our working dog work that we've been involved with, and a little bit in the second part of my talk about reproductive genetics, which is really exciting new work that's been happening in our lab. Oops. So we'll start the first part of my talk today talking about the genetics of farm dogs, and a project that's been going on funded by the Rural Industries Research and Development Corporation in Australia and the Working Kelpie Council of Australia to better explore the traits that are important to people who have livestock herding dogs in Australia to work with sheep. And the main breed we've been focusing on is the Australian Kelpie, which is a breed that was developed in Australia, and I'll tell you a little bit more about that in a moment. In the second part of my talk, I'll talk about some work that we've been doing with field trial Labradors and also some reproductive behaviour work. So to start my talk, we'll talk about beginning a selection program for the working Kelpie in Australia. Working Kelpies are very widely used in the rural areas of Australia. It's estimated that there are about 40,000 working properties in Australia, and each of those has about 3 working dogs, and usually this is the breed that's most popular, is the Australian working Kelpie. But there is a problem sometimes where dogs are not placed correctly in the correct home, and this leads to dogs being lost from the industry as being unsuitable for the work that they do. And our goal is to try and improve the rate at which dogs are successful in their working life so that they can live a long and happy life with their owners and in their working situation. So to start out, the first thing we needed to do was to figure out what are the things that are really important to the livestock farmers who are using these dogs. And that's actually not as easy as it seems, because even though different farmers or handlers might say that they're interested in a particular trait, when they say that trait, they may not mean the same thing as a different person who's discussing that trait. So if it's farmers talking about that they're very interested in the trait of cast, which means where the dog runs out and runs around the sheep to bring them back to the farmer, that might mean something different to that person than it does to another person. And so the first thing we needed to do was to figure out which of those characteristics were the most important ones so that we could concentrate our attention there. The next thing we had to do was to figure out how to assess the different phenotypes. Phenotypes being the traits that we're interested in. And that's not as easy as it seems, because if you think about it, these dogs are dispersed all around the countryside. They're not just in one place. So that makes it very hard for us to bring them into a test situation that might be used in as was used in the studies that some of you might have seen if you joined us yesterday. So instead we had to figure out a way to assess these dogs remotely, if you like. And to do that, we tested a few different avenues, which we'll talk about in a moment. So we wanted to use these phenotypes, these traits of interest. We wanted to help the breeders to concentrate their breeding to improve the traits that were important to everybody, And then we wanted to reduce the any welfare issues in the breed. So there's two ways of looking at this problem of how much genetics impacts the actual working ability of the dog, because as some of you may have learned if you joined us yesterday, most of the traits that are of interest to us aren't just genetics, they're also the environment of the dog. And by the environment, I don't mean whether it's sunny or rainy, or whether there's grass or not. What I mean is whether the owner has other dogs on the property, whether the owner is an experienced dog trainer, the personality of the owner, the age and experience of the dog, the background of the dog. All of these things work into the environmental effect that confound the results that we're working with. So we never expect genetics to give us the entire answer. It will only ever give us a part of the answer in this context. The top-down approach starts from working at the level of the traits and working down to figure out what's happening genetically or to make genetic changes. To do this, we use statistical genetics techniques. So we use a thing called estimated breeding values. To use estimated breeding values, which are trying to estimate the transmissible genetic superiority of the parents, we first need to figure out, for our important traits, how heritable they are and how they connect with one another. So if I'm selecting for caste, will that have a negative effect on something else that we're interested in? Eventually, we want to make a resource to calculate estimated breeding values for these animals. That's probably some way down the track yet, but we're working in that direction. So the first step in our top-down approach is to ascertain the traits that are of importance to breeders. And to do this, our PhD student, Jonathan Early, went to the literature and he purchased, or we obtained from the library, several books that related to the breeding and training of working kelpies or working dogs in Australia. And a text search was conducted to identify terms that were of interest to the people that had written those training manuals for those dogs. And those terms were collected and counted how many times they appeared in the document that was being written. And so here we have a collection of the behavior or the personality traits, if you like, of the dogs that were identified as being most important for these dogs. So they were confidence, whether the dog was keen, its temperament, if it was willing to work, how strong it was. And in that context, they're talking about not strength, but how mentally strong, how resilient the dog is to put up with adverse conditions in its environment, how obedient it was, how intelligent it was. And then if it was weak, that's a negative. This one is negative traits. So they don't want a dog that's weak. So it's important that we don't use dogs that are weak in the same sense that we're talking about strength before, or that are fearful. And we want dogs that are calm. We don't want dogs to be out there and harassing the sheep. But in all of these things, the very most important one you'll see is the first one, which was confidence. They wanted a dog that was unbothered by things, that could focus on its task and do the job at hand. So while these were personality traits, we also identified different working traits that were important in those manuals. So I'll explain each of these as I go. So the first one that was most important was a term called caste, and I did actually explain that earlier. So that's where you send the dog out into the field, and in Australia, the ranches or farms are extremely large, and the dog might be asked to run over several hundred acres to what we call muster a flock of sheep that is dispersed through that paddock. And their goal is to run around the outside of the sheep, to gather them together and to bring them back to the owner. The next trait is one called head. And what head means is that most of us think about when we're moving animals that we drive them from behind. But the difference with the way these dogs work is they work at the front of the sheep to bring them to the owner. So while the sheep might be trying to get away from where the owner is, the dogs will work at the faces of the sheep to bring them back to the owner, who might be standing at the gate and just waiting in the field. So the owner might just say, "Way back, Scruffy," and Scruffy will run out around the back of the sheep and bring them in, to the farmer who's standing at the gate. And I can tell you a funny story about that, because when I was an undergraduate student doing work experience on my first working property, on a sheep property, I was asked to mind some sheep on the long paddock, which is the roadway. And it was during a drought, and this farm had a lovely paddock of alfalfa or lucerne right next to the road, and the sheep got into the paddock. And I had been given 2 working dogs to work with, and I'd never met them before, and I'd never worked with working dogs before, and I was asked to bring the sheep in. And I kept trying to go out and run around the back of the sheep, and it took me some period of time before I realised all I had to do was stand at the gate and the dog would bring the sheep to me. Anyway, so where was I? Cast head, bark. So most, not all, working contexts for livestock herding dogs require silence. They don't want the dogs to be yapping at the sheep. There are some working contexts that do desire bark, and this creates a divide in the way the dogs are used. So it's really important that the right dog is used in the right context. So if you're pushing animals up a livestock race at a sale yard or onto a truck, then a bark might be desirable. But if you're working the dog out in the large field, you don't really want a dog that's barking. The trait of eye is where the dog crouches and uses what we call at home the Jedi mind trick to control the sheep. So it knows the correct working distance, it controls the gaze of the sheep and manoeuvres the sheep using its gaze on the sheep. Then we have force, which is the correct working distance from the sheep for the context in which it's working. So you don't want a dog that's either too close or too far back. You want a dog that is close enough to have the sheep listen to the dog and manoeuvre to the place they need to be. Then we have hold, which is the ability of the dog to hold the sheep in a particular location. And then we have balance, which again relates to the correct working distance from the sheep. Drive, which is their desire to work all day long, which they often have to work, they cover many kilometres or miles in their day of work. Their ability to control the sheep, and cover, which is their ability to make sure that no sheep escape, that they're covering the entire flock, that they're not letting sheep drift off the side of the flock. So these are all things that most of you have probably never heard of, but are really important in the working context in Australia. So as I said, we collected these terms from the books, and the next thing we had Jonathan do was that he had a number of owners and handlers of working dogs assess videos for the dogs for these different characteristics. And then we had a group of experts that we identified through the Board of Management of the Working Kelpie Council. So these were all very, very experienced working dog breeders and livestock herding handlers. And we had them assess and score dogs on a series of videos for these different characteristics that were ascertained as being important. Next, we did a statistical analysis to see how well those two groups agreed with one another, because our goal was, in the end, we needed to be able to test these things remotely. So what we wanted to be able to do was to ask the owners to assess their own dogs. We had to ensure first that they were able to do that properly. So I apologise that this slide is a little busy, but at the end of the day, the start items are those that were in our top 10 items for working manoeuvres. And the end results were that if the average handler scored the dogs and the group of experts scored the dogs, the agreement within the two groups was pretty much the same. So you couldn't say that you needed the experts to score the dogs, because really everyone was doing pretty much the same job of doing the scoring. So we had similar levels of agreement between the scoring of these traits on the videos for the different groups that were being used. So at the end, we designed a survey tool to assess these dogs remotely, and we invited Elizabeth Arnott and Jonathan Early, travelled out into to the country areas and met with different people who were breeding and handling livestock herding dogs, and had them fill out, in their first instance, a paper survey, but was later put online. And if you Google farm dog, and the University of Sydney will probably take you to this page if you're interested to participate. Since then, we're developing a different web resource, which is a little bit easier to handle if you have multiple dogs, but we're still using this one just for now. We also invited the survey participants to donate DNA from their dogs to our project, and we collected that in our lab, and we've then sent that away to have genetic markers ascertained on those samples. So for some of these dogs, not all of them, we have corresponding behaviour surveys and DNA samples. It's actually harder than you'd imagine to have the survey and the sample line up with one another. It's often easy to get a lot of DNA samples and easy to get a lot of surveys, but it's hard to get the exact same dogs to do both things. With the phenotypic data, so with the survey data that were gathered during this project, we were then able to ascertain how heritable these different characteristics were. And what we found, and we asked the question of how related dogs performed in these exercises, using in both situations that included the presence of stock or where stock were not there, so stock being the sheep or the cattle. And what we found that were moderate heritabilities, although at this stage the standard errors which are reported on the right are quite high because our sample size is relatively limited. So we can, even though this gives us a suggestion that these traits are heritable, and we know they must be because they've been selected in that line and they've made improvement, and clearly those Kelpies are different than, say, your average, Labrador Retriever or Golden Retriever or Dalmatian. We know that they're heritable, but you can see from the levels there, heritabilities are the proportion of the genetic superiority of the parental group that's exhibited in the progeny. And you can see that the value should go between 0 and 1, and you can see it's about 20 to 30%, which is sort of what we'd expect. And that's not a bad figure, actually, and that's definitely something we can work with. So that's where we're up to with the phenotypic part. So that's just using the survey data. At that stage, we've not done anything with the DNA. So that's what we call the top-down approach. Next, we're interested in learning more about the bottom-up approach. Can we look at the DNA and figure out which parts are important to the different livestock herding breeders? Because it may be that the breeders think they're selecting for something, but actually they're selecting for something else. And can we actually see which parts they really care about? Now, those of you who were with us yesterday during Chris's talk might have heard him use a term called the footprints of selection. And so what we wanted to do was to compare two different groups of dogs to see if we could figure out the places in the DNA where those two groups of dogs differed. Many of you might be unfamiliar with this breed, the Australian Kelpie. And even if you're familiar with the breed, you might be unfamiliar with the idea that there might be two versions of this breed. Back in the early 1900s, this breed was basically, it was developed in the late 1800s. I'll talk more about that in a moment. But in the early 1900s, it began to be divided. We had a group that became show dogs and pet dogs, and we had a group that became working dogs. And they began to be registered by two different registration organisations. The working dogs were registered by the Working Kelpie Council, and the show dogs were registered by the Australian National Kennel Council. This provided us an opportunity to compare the DNA of these dogs, because this split, there was no requirement for the show dogs to do work. So this provided an opportunity to see if there was anywhere where the working dogs had particular differences in their DNA that were not present in the show dogs. So to do this, we used a method called a selective sweep analysis. So in a selective sweep, if you can imagine here, we're showing on the left is a picture. These are actually human pictures. Well, they're not really humans. They're from a diagram, obviously. But here we can see representations of different chromosomes for different humans. And you can see that there are different areas that are light and dark in each thing. And these represent different backgrounds of DNA or different varieties of genes, well, the same gene, but different versions of the gene that might behave slightly different in a different environmental context. Before selection, there's a great variety of chromosomal patterns that are there on the left here. But you can see that after selection, what happens is a certain pattern which is desirable in the population will be selected for. So here, in this population, this pattern was quite rare. But this one was the desirable one that was taken forward, and you can see that it's highly enriched in this new selected population. So what we wanted to do was to check the DNA for the show and pet kelpies, and to check the DNA for the working kelpies, and to see if we could identify a place like this where the working kelpies had an enrichment for one particular type of pattern. And the way we would see that would be that we would see only one kind of marker there. We wouldn't see so many of the other kind. So at this stage, what we did was we collected all of our DNA together. We sent it off to the laboratory. We don't actually do the measurement of the markers in our laboratories. It's a specialist operation that's done elsewhere. We collected that back in. Jonathan Early and Elizabeth Arnott went off to a dog show and collected some samples from some dog show kelpies, and we brought those back. And then we did the same process for them. We sent their DNA away to the laboratory, and we got back from the laboratory some data files that contain the marker results for the different animals. So then the next thing we did was we compared the markers for the show kelpies with those from the working kelpies at about 170,000 different locations across all of the chromosomes of the dog. And you might have seen yesterday that the human chromosome has 23 chromosomes. Sorry, the human genome has 23 chromosomes, but dogs have many more chromosomes. They have 38 autosomes plus the X and Y chromosome. So we were interested in places, so typically where there's the types of marker that we're looking at, typically there are only two kinds possible. So we were looking at places where there was only one kind there in the particular group that we were studying. And we call that, where there's only one kind, we call that variant an allele. And the minor allele is the one that's the least frequent allele. So where there's only two, there's a major one and a minor one. So we were looking at places across the genome. We averaged the minor allele frequencies within particular sections of the genome, and then looked for places where this was persistently low over a contiguous or connecting portion of the DNA. So let me explain this chart because it's a little busy. In this chart, it's a little bit smooth to make it easier to view. It's quite jagged if you look at it in its raw form. But what we're showing is the minor allele frequency across the whole DNA for a kelpie, different groups of kelpies relative to our reference genome sequence, or the basic genetic map that all of us work with, which is a boxer sequence, which is kind of not really relevant. It just tells us where the different genes are. And in the blue line here, what we're looking for is a place where the blue line goes down really low, but the orange line, which represents the show kelpies or the pet kelpies, stays up high. And we could do the opposite thing too, and we might talk about that again in a minute. So, but for the working kelpies, there was only one place, there was only one single place in the whole DNA where this happened, and that was here on chromosome 3. And in this place, you can see that the orange line stays high. So on the side here, we have the minor allele frequency. We can see that across the group, the minor allele frequency across the genome is about 14%. So that means that even though people might think that purebred dogs are all homogeneous, this tells us that in any average location, there's about 15% of alternate alleles there. That varies, of course, from place to place, but in this particular instance, here was a place where the working kelpies had very low variation, and the tipping point for us in this study was for it to go below 5%. And this might have allowed for a margin of error, but it also allows for the case where there's the odd recessive allele that no matter how hard they select, they never get rid of it. And so this region here on chromosome 3 is very big. It goes for about 8 million letters. It's a huge area in genomics terms. And it was very interesting us to see then, well, okay, so here we've got this region where the working kelpies are considerably different than the show kelpies. What's living in that region? What can it tell us about the way these dogs are selected? So here's the region of interest. For those of you who are not geneticists, this is going to be a little bit of a leap, probably. But what we do, we use the power of comparative genomics to tell us more about what's happening in that area. And so the genomic resources for dogs are very well developed, but they're not as well developed as are the resources for two other species, and those are humans and mice, and in particular mice. And there's a particular facility that has been lovingly constructed by very devoted scientists over a very long period of time to not only tell us which genes live in which places on the DNA, but also to tell us what those genes might do. It might surprise you to learn that there's a lot of similarity between a dog and a mouse, as it is between us and a dog or us and a mouse. And so what we can do is we can take that region from the dog DNA, we can look at the genes that live in that region, and then we can look for where those genes are on the mouse genome, which is what we've done here, and we can see if the genes are in the same order there, which tells us that it came from the same common ancestor way, way, way long ago when we were all squirrels in Ice Age. And so then we can see what these different genes do. So here we found a big region. Actually, this region here is only 3 million base pairs in the mouse, but it contains all of the same genes that were in the deepest part of the selected region in our working kelpies. And when we look there and we see what these different genes do, it gives us some idea of what they're involved with. So for example, we have genes that are involved with fear conditioning. We have genes that are controlling nociception. And you might have, for those of you that watched yesterday, you might have heard, I think it was Chris talking about dogs' ability to not feel pain. Nociception is our ability to sense pain, particularly in our extremities. So can you feel heat or cold? And so, and we also had things on relating to eyesight. These two relate to eyesight. This one relates to gait. And kelpies do have a very particular way of moving, where they tend to work in a crouched form. And then we had some others over here that also related to the ability to feel pain or anxiety. And what this was fascinating to me, because this was the sole place in the DNA where all of these dogs had the same thing. And if you talk to any livestock breeder of these breed out there, they would tell you that, oh, it's really important, you need a heading dog, or you need a dog that has an apple-shaped cast, or you need a dog that's fearless, or you need, well, that probably does relate, but you need a dog that has all these particular characteristics that they believed that they were selecting for. But in the end, what it came down to was the fact that if your dog cannot, what you might not understand is in the outback areas of Australia, you cannot walk in bare feet. So if you walk out into the paddock with no shoes on, paddock is a field or the area where the sheep are grazing, what you'll find on the ground there is a very, very large number of prickles and spiny species of plant. And you can imagine if you're a working sheepdog and you cannot walk out into that field of prickles, you can't even begin to herd the sheep. So this was an amazing idea of an amazing thing where the data tells the story way better than what we as humans believe we're selecting on. So while the humans thought they were selecting on these working characteristics, actually what they were selecting for was the ability to run on prickles and to maybe not remember fearful events that happened to the dogs. So if the ram had, butted the dog, or if the dog had had a bad experience, these dogs were resilient to that bad experience and were able to carry on. So these are the kinds of prickles that live out there. We call them bindies. I think you guys have another, you have them here, they come from Mexico. I think you have another name for them. They're quite nasty. They can actually puncture your footwear if you're wearing shoes out there as well. And these are the sorts of things that dogs have to run on. But there are other species too that are equally nasty. There's this one here that we call khaki weed, which has spikes on it. And there are all sorts of Scotch thistles and so on. And so these dogs were able to run out with their pads onto these fields and focus on the task at hand of herding the sheep, not of running on those prickles. I can see we've got a little bit of time left in this. I was going to finish up there, but we've got a little bit of time left. So I'll just go back and tell you a little bit actually about the other group of kelpies, because they had some things that happened with them as well. So just bear with me for a moment while I reverse. So you might see that there are a couple of places here where the show kelpies, where the working kelpies are high and the show kelpies are low. And so we have a place here on chromosome 8 that goes below our cutoff zone. And there was another one over here on chromosome 16 that didn't make the cutoff, but actually does equate with a phenotype that we observe in the dogs. In this site on chromosome 8, we're not really certain of what it is in particular that's there. The genes that live in that spot have mainly to do with skeletal formation. So it's likely to do with a difference in the structure of the dog that has been selected in the show dogs versus the working dogs. But on chromosome 16, there's a gene there that relates to coat colour. And that gene is, we know it colloquially as the K locus. And the K locus, it's always been thought that the black and tan phenotype has always been allocated to the A locus, which is on chromosome 24 here. The show kelpies are not black and tan or brown and tan. They're just solid black or solid brown. And the show and the working kelpies always have the tan points like the dog that was in the cover slide. Not always, nearly always. And so there's a very strong sweep there where I guess the show people wanted their dogs to look identifiably different than the working dogs. And so they'd selected for this solid type. But what this also tells us is that this, I identified in a previous study that black and tan versus solid coloured is controlled at this chromosome 16 locus as well as at the chromosome 24 locus. So you need a specific combination at both places for that to occur. So those are some interesting things from the conformation dogs. Sorry, I'll go forward again. So the Kelpie breed originated about 1870, and they were developed from what were called working collies. And I believe that these dogs were more like the smooth collie as opposed to like the lassie collie type rather than the border collie type. And they were smooth collies, but they had very little white marking. And these came from Scotland. They were already good working dogs when they were imported, and they were identified as being extremely good working dogs even back then. So really we can thank the Scottish for developing the great working characteristics in this breed that were just enriched by the selective maintenance of those characteristics in this particular Australian breed. But there was one particular female who came from a black and tan line, and she came into the possession of a guy called Mr. Gleeson, and he called her Kelpie, and she became known as Gleeson's Kelpie or Old Kelpie, and this is where the breed name came from. So that dog was then given was bred to another male that was owned by Mr. Mark Tully called Tully's Moss. He was a solid black dog, and that pair of dogs produced another bitch called King's Kelpie, and she was an extremely successful dog in the sheepdog trials of the time. And this is a picture of King's Kelpie. It's actually, well, it's probably a painting rather than a picture. That's from the history of the Kelpie. Here's actually, I guess it's a daguerreotype or something, of another very famous Kelpie in the Kelpie history. So the entire breed was based on this initial pairing of dogs. And there's quite a lot of line breeding on these dogs at the time. But eventually from this line of Kelpies was produced this dog who was called Coil, and he was a blue dog, so the dilute version, gene version, has been in this breed for quite considerable time as well. So back in about 1879 or something, there was the first ever Sydney Royal Easter Show, which if you're from Australia is a pretty big deal. And they had one of the first sheepdog yard herding trials there. And this dog, the trial was run over 2 days. And this dog ran on the first day and he did really well, he won. But sometime later that evening he had an accident and he broke his leg. And the next day he had to compete in the finals, and despite having a broken leg, he competed in the finals and he actually won it. So this plays into this idea of this ability of these dogs to overcome pain to undertake the task that they're really interested in performing. Here's another image, I'm not sure whether it's a photo or whatever, of an early show Kelpie in 1908. This is from The Australian, which is a newspaper in Australia, from Saturday the 9th of May 1908. And this dog with its fancy collar is Mr. A.G. Sewell's Kelpie dog, Berkshire Bosca, and won at the Sydney Royal Easter Show. And you can see already that there's a distinctive phenotypic difference occurring between these different lines of Kelpie, even at that early stage in the proceedings. So this, for me, we have funding for this project. We've had funding since 2015. We currently have funding for the next 12 months or so, but the goal is to make this an ongoing resource to work with these dogs. We want to create a resource that helps livestock herding dog breeders to better identify suitable working breeding partners for their dogs. And one of the big problems in the industry is that a lot of the breeders feel they're not well connected. Well, they are well connected, but they don't have a good concept of the quality of work that's undertaken by dogs elsewhere outside of their local regional area. As a result of that, there's a lot of line breeding that goes on in these breeds, and the breed is not devoid of problems. We have another problem, another project involving this breed that involves a cerebellar abiotrophy, which is a movement disorder in these dogs that can be quite debilitating. It's inherited, appears to be recessive. We actually think it's complex. And so we want to give people the best opportunity for identifying other breeders that share the goals of a particular breeder's breeding program. One thing that I've mentioned kind of in passing, but not precisely, is that there's not just, even though we've got this split between the working Kelpies and the show Kelpies, there actually is more than one kind of working Kelpie, which I've sort of alluded to, but haven't really elaborated on. The first kind of working Kelpie is what's called a paddock dog. They're the dog that you will take to muster the mountainside or the steep gullies where you can't get with a horse or a car or a bike. You have to actually have these dogs to work that area. And they say that, you know, a working Kelpie on a farm is as good as an extra 2 people working there, because they can save the salaries of that many people by doing the same amount of work that those people might do. So that's the paddock dog, that's the first kind. The second kind is the yard dog, and I alluded to those before, and these are dogs that are required to have a lot more force, a lot more bark. They occasionally they don't mind if they bite the stock. They might use those on cattle rather than sheep, and because cattle are much bigger, obviously, and require a much more forceful dog to manage them. And then the third kind are the trial dogs. So these are dogs that are bred specifically for doing the sheep trials that you might see on YouTube, where they have the 3 sheep and the little gate, and they have to whistle the sheep through the gate or up to the lounge to get their Guinness or whatever the beer commercial is that they have. And so that's a particular, the skill sets required of these dogs are quite diverged from one another. In the case of the paddock dog, you need a dog that's extremely independent in its working ability. That dog needs to be able to go out there, you give it a single command, it will go back, it will do it for you, it will figure out the best way to get the sheep, it will figure out how to get them back to you at the gate. It does it all. That dog will work independently of you, does it all but on its own. And those dogs are very valued. The yard dog, as I've said, needs those more forceful characteristics, but it's working in closer proximity to the owner. So it still needs to be very obedient, but it doesn't need to be so independent. So it can take direction from the owner. It's not really, and those, oh, sorry, when I said back before, I just realized that I said the wrong thing. So back is when the dog jumps on, you might have seen pictures of the kelpies jumping on the back of the sheep. When you've got a lot of sheep in a race and the dog needs to get from one part of the yard to the other quickly, they need to leap over the sheep. And sometimes they want to move the front sheep forward more, so they jump up and they harass the front sheep up to get them up the race, and that's called backing the sheep. So they need those more forceful characteristics. The trialling dog needs to be very attuned to the handler. It needs to work very closely in and listen very carefully and be very responsive to the needs and requirements of the handler. It's very different. These are all very different personality characteristics that are required in each case. Finally, there's a group that they call the utility kelpies, and these are the poor dogs that are asked to do every single thing. That's the person who has one dog, and they want them to be able to muster the paddock, put the dogs on the truck, and then go to the trial on the weekend. So a newer study that we've done has looked at whether we should be able to breed these different dogs from the same working kelpie breeder. And so in a more recent study, what we've done is we have gathered the information about these different types of working kelpies to see, first of all, are the personality characteristics required of them really different? Because people think they are, but there's never been documented. And are they genetically the same or genetically different? So when I say genetically the same, I mean, can you pool all the breeds, all of the different types together to do a mapping exercise to identify the genes that relate to those tasks or not? Or do we have to treat them as separate populations? And we've just had an article accepted in Canine Genetics and Epidemiology, it's not published yet, that actually addresses this question. And what we found was that indeed there are significant differences in behaviour types between the different working contexts. And this means that it's really important to put the right kelpie in the right home to make sure that it has the best chance of success in its working life. But we also had a small number of animals for whom we had DNA available. So we had, as I said, it's not always possible to get the phenotyping of the dogs or the survey scores of the dogs matched up with a DNA sample, but we have that for 80 or so dogs. And what we were able to do was to look at the DNA. We didn't do any mapping because really we're a little underpowered for that. But what we were able to show was that even though these are starkly different behaviours that these animals are exhibiting, they do all come from a homogeneous population. So we can actually do trait mapping in that population when we get the sample numbers that support that. And of course, if you say this to a working kelpie breeder, they say, why do you bother doing that? Why are you telling us this? We know this. We know this. And I have to say to them, well, yes, you know it, but the scientists don't know that. It's not documented in the literature anywhere. Someone who comes along later to study this wouldn't have ever known this unless we'd done this work. So it's really important to set up these base studies to actually set the foundation for the work that follows on. And where we're up to now is we're continuing to collect surveys from animals. We're trying to create the estimated breeding value resource. And we're still collecting DNA samples from different dogs. In fact, we've worked with the Working Kelpie Council to identify elite working dogs from their perspective. And we're making a special set of DNA of those dogs that we will then use as the standard to which we compare the other dogs for their abilities. And I didn't put the pictures on here, but we have some nice little charts that are able to show people where their dog's ability sits in relation to the average dog and in relation to the elite dogs. So that's all very important work that has been done so far. So this work is still underway. We have a new website that's not live yet, unfortunately, that we're going to use to enable people to manage several dogs at once on their site and to bring data in. Hopefully that resource will be an ongoing resource in the longer term. And we hope to really be able to help people to, first of all, identify how good their dogs are in relation to others. And people often say to us, how are you going to do this? Because, you know, this person, this handler, they don't know as much about herding as I do. You know, it's not going to be accurate. The way we make it accurate is not through looking at individual dogs themselves. We're interested in the relationships between dogs. We're interested in assessing the parental dogs. We're seeing how their progeny perform. And then we can relate to the genetic potential of the parent dog by looking at how well their progeny are performing in different aspects of the things that we're interested in. So that's about it for the first part of my talk this morning. I'm going to wrap up now. This afternoon, well, sorry, not this afternoon, but in the next part of the talk, I'll come back and we'll talk to you more about some other work that we're doing in relation to mapping traits that are important in elite retrieving dogs, and also talking to you about traits that are important in relation to the way that dogs breed or reproduce, which is a really important characteristic for many aspects of canine biology. So I'll leave it there, and I'd love to thank you for joining us. Thank you to you guys out there. Please post your pictures. I want to see all of you at home joining in. The Toller people out there, hello, and I'd love to see you tweet in as well. The people in Australia as well, thank you for joining us, and those of you around the world. It's been wonderful talking with you today, and I'll just wrap up there, I think. So thank you.